Hello everyone, welcome to Unit 1, which is going to be all about molds and yeasts that appear in our food. So, first off we're going to be talking about yeast, which are a type of unicellular fungi, which means it has one cell. It's not going to be composed of multiple cells. And so, some of these are going to be producing something known as pseudohyphae, which is these filaments that are very similar to hyphae, but they are not actual hyphae, they're pseudo-fake hyphae. So then our life cycles, we have two different ways of reproduction, asexual and sexual. For asexual, there are two different ways. There's budding and fission. So budding is where you have the larger parent cell and then a smaller daughter cell is budding off of that. Whereas with fission, you have a parent cell and then that splits into two equal daughter cells, okay? And then we have sexual, which is going to be through ascospores, okay? Right here we can see the life cycle. So I'm not going to ask you to memorize this or anything. Just know that the sexual reproduction is ascospores. These are asexual, okay? Now then for yeasts, we have a couple different kinds, and they're just based on their characteristics, so whether they ferment or if they're oxidizing film yeasts. So our fermentative yeast, so they're going to be facultative aerobes, and they're going to include Saccharomyces cerevisiae, which is a pretty important one. And so what that is going to do is it's going to convert the sugars, and that's going to change it into CO2 and ethanol. So this is very important for creating alcoholic beverages and then also with bread. So when making bread and the bread is rising, what causes it to rise is the CO2 being produced by this fermentative yeast, the baker's yeast, and then the ethanol will eventually evaporate once you are baking the bread, so you don't have an alcoholic bread. So, baking and brewing have both been around for a long time. They were pioneered in ancient Egypt around 4,000 years ago. And so, when brewing beer and wine, in order to keep the ethanol in, what they would do, and what we still do, is we cap the beverage so that the ethanol does not evaporate, okay? So we put a cap on top of the beer barrel or wine barrel, whatever we're making. That's going to keep it from evaporating out. And as you know, that's not something that we do with bread. Most commonly used yeast is, again, Saccharomyces cerevisiae. Very important. So, for beer fermentation, we have two different types we're going to talk about. We have ale, which is going to be a top fermentation, so it's going to be taking place up here, and that's going to require a warm temperature. And then we have bottom fermentation. It's going on down here. You can see a little foam. That's going to be requiring a cold temperature, okay? Then we have our film yeasts, and these are going to be causing a lot of food spoilage, so these aren't going to be nearly as useful as our fermentative yeast. So these are going to be strictly aerobic. They're going to cause spoilage of juices, brines, and wine, and they get their name because they produce a film on the surface of the liquid. So an example of this would be Candida mycoderma, and this is known as a wine flower. Okay, so this is not something that you'd want in your wine. So ethanol yield will be reduced. So for this one, we have our growth characteristics of yeast. So the maximum specific growth rate, so the higher the growth of the organism is on this axis, the y, and then the temperature. So as the temperature increases, you can see it increases up to a certain point, and then starts to decrease from there rapidly. So we have two different strains. We have S. cerevisiae and S. kudryasvae. Okay, so the circles one is here, and then cerevisiae is here, okay, with the squares. So we can see that S. cerevisiae, which is one of the most thermotolerant or heat-tolerant yeast, and it's going to be able to, it's going to be able to survive over 45 degrees Celsius, and then its optimal temperature is between 30 and 35. So our molds in food. So molds are going to be a filamentous fungi. Okay, so they have hyphae. They have actual hyphae. It's not pseudo-hyphae. And so these are going to be useful in food production, and they're also going to be involved in food spoilage. Okay, so a couple of foods that are produced using mold are cheese, soy sauce, and tempeh. So white mold cheeses. First one would be brie, which is also referred to as the queen of cheeses, and this is going to be coming from Penicillium candidum and Penicillium camemberti. Then we also have camembert cheese, which will be strictly from Penicillium camemberti, okay? And this is named after the village in France. Then we have our blue vein cheeses, which are going to be named based off of the region they're from and the milk used. So Roquefort is in France, Gorgonzola is in Italy, and Stilton is in England. And these are going to be using Penicillium roqueforti and Penicillium glaucum. Then we also have soy sauce, which originated in China approximately 2,500 years ago. So the fermentation of soybeans and mixed grains is going to produce this liquid with the assistance of Aspergillus oryzae and Aspergillus soyae. And then we also, of course, have spewed foliage caused by mold, as I'm sure you've all seen on your fruits and vegetables and bread that you leave out for a little bit without eating it. So outdoor mold just in the environment and the air around us has approximately more than 10,000 spores per cubic meter. And molds can grow on lots of different foods, as I'm sure you know, and can be very colorful. So Saccharomyces cerevisiae is a fermentative yeast, as I talked about a little bit, and it divides by budding. This is going to be one of the top fermenting yeasts, brewer's and baker's yeast. Then we have Schizosaccharomyces octosporus, which is going to divide by fission. It also has asci with ascospores. Okay. So it's going to be also fermentative yeast, and it will divide by fission. Okay. It's a little bit different than the budding. Then we have Candida cruzae, which is a film yeast, and it's going to divide by budding and has the presence of pseudohyphae with these blastoconidia. So this is going to be used to ferment cacao beans along with fungus Geotrichum. That'll help to eliminate the bitter flavor in chocolates. Okay, so just for a little bit identification of molds. So here in the very top we have conidiospores. Down here we have septate hyphae. So you can see the septate from the septum. It's referring to these little lines. These are the cell walls in between these individual cells that make up the hyphae. And then here we have non-septate. So there's no septum, no cell walls separating it. It's just one large non-septate hyphae. Okay? Nothing to separate them. And then down here we have our rhizoids. So these are going to be for any of the rhizopus species. And then at the very top they're going to be producing sporangium. Okay? So now we have Aspergillus, two different species. We have Flavus and Niger. So Flavus is going to be yellow-green spores, and that's going to produce an aflatoxin. And then we have Niger, which is going to produce a black spore, okay? So you can tell them apart just because this is a more yellowish-greenish color, and this is going to be blackish. Okay, so again, these are going to have the conidiospores, conidiophore, okay. Then we have Penicillium, which we have two different species, Penicillium camemberti and Penicillium notatum. There's going to be white spores for the camemberti and blue-green spores for the notatum. And here we have conidia on the top, a conidiophore, which then connects it to the hyphae at the bottom. Then Rhizopus stolonifer. So now these are going to have the rhizoids, sporangiospores, sporangiophores connecting those to the hyphae, and then vesicles here where all the spores are going to attach, and the whole thing is going to be sporangium. So now we have Fusarium roseum, and the roseum is due to the pinkish color So plant pathogen, and it can also cause cereal and fruit foot rot. So it's an opportunistic human pathogen. So it can cause disease in humans in certain circumstances. Okay, so this is just a list of organisms that can be found and where they're found, how they look, and then what's their importance. So it's just for your information if you ever wanted to see a condensed list of everything all in one spot. So that's it. Make sure that you do everything that's on Blackboard. There's a couple other videos I want you to watch. You need to make the discussion board post and finish the quiz, but that's it. Good luck, everyone.